Hi, my name's Soraya Palencia, and the news article and primary article that I chose to do my presentation on is called Insights into the Regulation of the Expression Pattern of Calvin-Benson-Bassam Cycle Enzymes in C3 and C4 grasses. The news article starts off by saying that there is not much known about how the spatial expression pattern of other Calvin-Benson-Bassam or CBB cycle enzymes are regulated, but there is evidence that changes in the regulatory sequences of rubisco contribute to the bundle sheath specific expression. So in the experiment, the researchers used a computational approach to scan for transcription factor binding sites in the regulatory regions of the genes encoding CBB cycle enzymes, SBPAs, FBPAs, PRK, and GAP-DHP of C3 and C4 grasses. According to the news article findings, they identified potential cis regulatory elements present in each of the genes studied, regardless of the photosynthetic path used by the plant. The transacting factors that bind these elements have been validated in A. thaliana and could possibly regulate the expression of the genes encoding CBB cycle enzymes. They also found C4 specific transcription factor binding sites in the genes encoding CBB cycle enzymes that could potentially contribute to the pathway-specific regulation of gene expression. These results provide a foundation for the functional analysis of differences in regulation of genes encoding CBB cycle enzymes between C3 and C4 grasses. Compared to the primary article, the news article didn't have as much information, but it was still accurate. And the primary article specifies how this is the first study to focus specifically on the differences in the regulatory sequences of CBB cycle genes. And the primary article goes more in depth on the compartmentalization of C4 CBB cycle enzymes, which the news article doesn't address. So according to the primary article, there are conserved potential cis regulatory sequences shared between C3 and C4 species. However, this alignment approach is based on sequence identity over at least 50 BP, so it was possible that smaller motifs could have been disregarded. Then, using the same approach to identify TFBS absent from genes encoding C4 enzymes, revealed two C3 specific motifs in the 5' intergenetic region of PRK. These results suggest that there are C3 and C4 specific TFBS that might contribute to the compartmentalization of C4 CBB cycle enzymes. The researchers first determined the A. thaliana TFBS shared between orthologous genes and used those to compare between the genes encoding the selected 4 CBB cycle enzymes. They were then able to identify one TFBF present in all of the potential regulatory sequences that was bound by VRN1 in A. thaliana. This TFBS was also identified in the putative regulatory regions of genes encoding photorespiratory and housekeeping proteins, which suggests that it may play a regulatory role not limited to photosynthetic genes. So to get these results, they used two different approaches to identify potential regulatory elements that might contribute to the compartmentalization of CBB enzymes in C4 species. The alignment of the genomic regions of the orthologs encoding the selected CBB cycle enzymes allowed researchers to identify conserved non-coding sequences shared by C3 and C4 orthologous genes, but the scanning of putative regulatory regions with TFBS validated in A. thaliana allowed them to identify putative C4 specific regulatory elements. The results found provided new information on putative regulatory elements of the genes encoding SBPAs, FBPAs, PRK, and GAP-DHP in both C3 and C4 species. The fact that these CNs are only shared between the species of the PAC-MAD clade, including D. oligosanthes, which perform C3 photosynthesis, suggests that these CNs do not play a role in C4 compartmentalization, and instead they are a result of shared evolutionary history. And in the primary article, there were a series of figures and tables. And in Table 1, we're able to see differential expression of transacting factors binding putative TFBS in bundle sheath and mesophyll cells of C4 species. In Figure 1, the graph shows sequences with 50 to 100% identity and regions with greater than 70% identity within 50 base pairs are highlighted in purple if they are located in exons, in cyan if they are located in UTRs, or in pink if they are located outside exons or UTRs. In Figure 2, it shows the identified A. thaliana TFBS and in which orthologous genes they are found in. The horizontal bars represent the number of common motifs identified within orthologs, and the vertical bars represent the motifs shared between different orthologous genes. And this is my citation for all the figures and the news article and primary article. Thank you.